Hello students, we will be today studying about the contributions of Alexander Fleming. Now Fleming, he was from Scotland, and he worked as a medical doctor during the First World War. He also received the Nobel Prize in the field of medicine for his various contributions. So let us see what are his contributions. Now, basically, he worked, or his main work was in the antibacterial property. Now, what is the meaning of antibacterial? It means something that is able to kill the bacteria. So basically, he contributed in the antibacterial field. That is, he first and foremost worked on or he introduced penicillin, which is called as the wonder drug. And also he spoke about the importance of lysozyme, or he told, or he discovered lysozyme. He told where is this lysozyme present and what is the use of lysozyme. Moving on to the first contribution, that is the discovery of lysozyme. Now, what is this? Lysozyme is basically, it is an enzyme, and where is it present? It is present in the various body secretions. Like it's there in the tears, it is there in the saliva, in the mucus, in the sweat, all these places the lysozyme is present, also present in egg white. Now, what will this lysozyme do? It will kill the bacteria. Okay, it has a property to kill the bacteria by lysing the cell. It will lyse the bacteria and thus it will kill it. And this was the discovery that happened by chance. And he discovered, that is, Fleming discovered it, the presence of lysozyme in the year 1922 when he was working with a bacteria, and he discovered this. Now, how did he discover? Now, what happened was, when it was apparently, it is told that he was working with a bacteria wherein he was growing the bacteria in a lawn culture, that is, the entire plate was crowded with the bacterial growth. Micrococcus or staphylococcus, these were the organism that he was working with. That's the lawn culture, wherein the organism would grow throughout the plate. It was told, or apparently it is written that during those days he was suffering from cold, and by chance the mucus from his nose fell into the plate where he was working with the bacteria. And when he incubated the plate, and next day when he saw that there was a zone of clearance wherever the mucus fell. You can see in the first picture, figure 1, that the lawn culture, the bacteria had grown throughout the plate, and there is also a place in the center where the mucus had fell. Around the place where the mucus has fallen, you can see there is a zone of clearance, which means the bacteria failed to grow there. So he wanted to understand as to what is the reason that the bacteria is not growing. Then he took a little bit of the mucus from his nose, and he again inoculated into a fresh lawn culture, and he found that mucus drop did not allow the bacteria to grow, or in other words, killed the bacteria, and hence there was a zone of clearance around the mucus. Then he continued doing this experiment with in a slant, also in saline solution, where the microorganism was growing, and he added a drop of mucus to his discovery. He found that the entire turbid solution, that is, if the bacteria is growing, the solution will be turbid, but when he added mucus, after some time the turbid solution became clear, which means to say that the cells were killed by the presence of the mucus. Then he started finding out with the different body secretions like tears, then the sweat, etc., and he found that the microorganisms were killed. He later on found out it is because of the presence of this particular enzyme, which had the property to kill the bacteria, and he named the enzyme as lysozyme. Lys meaning to, lyso, meaning to kill the bacteria by lysing the cell. So that's why he named it as lysozyme. So that was his first discovery. The next discovery was he was working with the organism Staphylococcus. In the first picture you can see on the left hand side below, he was working with the organism Staphylococcus, and again he was growing the organism in the lawn culture. And while he was working with the bacteria, suddenly he had to go for a vacation. Again, it was written that he went for a vacation and he forgot to store the plate in the refrigerator, and he left the plate just outside. After few days, when he came back from his vacation, he found that the Staphylococcus was there growing on throughout the plate, but in one particular area he found that the Staphylococcus growth is inhibited, it has stopped, or there was a zone of clearance. And he found out that next to the zone of clearance there is one green colored patch, or there was some green color that was seen. Around the green color there was a zone of clearance where the Staphylococcus was not growing. And he again subcultured Staphylococcus, again grew the Staphylococcus on the plate, and he took that lawn or that green patch and he again inoculated over the Staphylococcus. He found that again this green colored organism inhibited or stopped the growth of Staphylococcus. Again there was a zone of clearance. Then he went on to isolate this organism, and he went on to isolate the component that was responsible to kill this particular organism Staphylococcus. He understood that this organism was releasing certain component or certain chemical or certain metabolite, we could say, and that metabolite was responsible for killing the organism Staphylococcus. He purified it, and thus this metabolite that he purified was named as penicillin, and the organism that released this chemical was penicillium. So this penicillium was the organism that was killing or inhibiting the growth of Staphylococcus, and this is the wonder drug which is called, because it is a broad spectrum antibiotic. It kills the bacteria by breaking the bonds of the cell wall. It basically destroys the cell wall, and thus the organism is killed, that is the bacteria is killed, and hence it's a broad spectrum antibiotic. It can be used for many organisms, many kinds of bacteria, most of the bacteria to be killed, against most of the bacteria. So thus the wonder drug was created, that is penicillin, for which he was awarded the Nobel Prize. And thus the first antibiotic came into existence, and that was given by Alexander Fleming. So these were the two main contributions of Alexander Fleming, which is lysozyme, which is which has an antibacterial property to kill the bacteria by lysing the cell, and another one is penicillin, which is released by the organism penicillium, again an antibacterial property which will kill the bacteria by destroying the cell wall of the bacteria. So these were the two main contributions. Thank you, students.